Next method used in the downstream processing more frequently is centrifugation. So the basic objective of centrifugation technique is also for the removal of insoluble materials present in the fermentation broth. So whenever filtration is not a suitable method, then centrifugation can be used as a method for the separation of solid particles. For example, microorganisms and other similar size particles can be removed from the broth by using a centrifuge when filtration is not a satisfactory method for separation. These centrifuges, which are used for the harvesting of fermentation broth in industries, are all operated on a continuous or semi-continuous basis because industries has to handle large volume of fermentation broth. Some centrifuges can be used for separating two immiscible liquids also, yielding a heavy phase and light liquid phase, as well as solid fractions. That means centrifugation is very useful for the separation of solids from the liquids, as well as it can be useful for the separation of two liquid phases. That means two immiscible liquids or the liquids with different densities. Now the principle of centrifugation, it works on a law. You need to remember this. This law is called as Stokes' law. According to the Stokes' law, the statement is this: the rate of sedimentation of spherical particles suspended in a fluid of Newtonian viscosity. Newtonian viscosity means homogeneous viscosity. Newtonian viscosity characteristics is proportional to the square of the diameter of particles. You need to read this again and again. Therefore, the diameter of solid particles means more frequently these are cells. It can be increased by flocculation or coagulation. That will increase the rate of sedimentation, which is a very likely logic that can be applied here. So during flocculation, there is neutralization of anionic charges which are present on the surfaces of microbial cells. That means in general we know that the overall charge present on the cells is negative. So that is a major hurdle, or it prevents the aggregation of microbial cells. So if these charges are neutralized, then microbial cells, they may aggregate to form bigger size solid particles, and it helps in their removal, or the rate of sedimentation will increase. Flocculation, it involves the mixing of process fluid with flocculating agents. These flocculating agents involve, examples are alum, calcium salts and ferric salts, tannic acid, titanium tetrachloride. Cationic agents are quaternary ammonium compounds, alkyl amines, and alkyl pyridium salts. All these are different types of flocculating agents. Now, there are different types of continuous centrifuges which are used industrially. These types of centrifuges are as follows. First one is the basket centrifuge, or it is also called as perforated bowl basket centrifuge. So it is responsible, or it is used for the separation of mold mycelia, that means fungal mycelia, or more crystalline compounds. It is a perforated bowl that means it is a vessel, and it is lined with a filter bag of nylon or cotton, and it is operated at 4,000 rotations per minute. This is the design of that basket bowl centrifuge. You can see this is the rotor, which is having a shape of basket, and it is rotating like this. It is internally it is covered with a filter cloth, and it is a perforated bowl, and it rotates like this. So whenever it rotates like this, so liquid is thrown out of this bowl, and it is forced to pass through this filter cloth. So whatever the filter cake is there, it is built up inside the basket, and the filtrate or the clear liquid, it is thrown out of this basket like this. Second type of centrifuge, which is operated continuously, is the tubular bowl centrifuge. The name of this centrifuge is because its rotor, you can see the rotor, that is A, it is like a tube, and that is why it is called as tubular bowl centrifuge. You can see the shaft at the center, that is indicated by H, then G, which indicates the rotor, and other parts like E, D or the components. You can see B, it is the shaft which is attached to the rotor, or it is attached to the motor which rotates this tubular bowl. Then this is heavy liquid outlet for the heavy liquid phase out, and this is the outlet for light liquid phase out. The advantage or the function of this tubular bowl centrifuge is that it is useful for the separation of two liquids having different densities, and therefore this centrifuge, it is operated at very high speed, a speed which is sufficient enough to separate two liquids having different densities. So that is the advantage of this centrifuge. But it is operated with broths, fermentation broth, which contain less than 10% of solid in the slurry, and it is one of the sensitive centrifuges. So the particle size, which ranges from zero point 0.1 to 200 micrometers, so this range of particle sizes can be separated by using this centrifuge. So this centrifuge is intended for the use for separation of light liquid phase from heavy liquid phase. It can be used for separation of solids from liquid liquid phase and heavy liquid phase separations. It is also useful for separation of solids from liquids. So it has multiple uses, and which is one of the most frequently used centrifuges in the industries. It is operated at very high speed, that is 50,000 rotations per minute. You can see this is the rotor which is rotating like this, and whatever the liquid which is there, which is feed in from here, this end from the bottom, it will enter inside the rotor, and if it is a separation of two liquids, then the light liquid, it will gather at the center of this rotor, and heavy liquid, it will be thrown out, and it will be gather around the periphery of this rotor. You can see here at the center there will be light liquid, due to centrifugation, centrifugal force, and at the outside, means at the periphery outside of this rotor, there will be presence of heavy liquid phase. So this red arrow, it shows the separation of light liquid, which will be at the center. It will be separated from this end, and there is a separate outlet for the separation of heavy liquid, which will be found at the periphery of this rotor, and it will be separated from this end. So this is how light liquid phase and heavy liquid phases are separated using this high speed centrifuge. This is again a little simple diagram which shows the separation. So yellow color, you can see yellow color liquid is light liquid phase, which is gathered at the center, and heavy liquid fraction, it is at the outermost part or along the wall of the rotor. Third type of centrifuge is the solid bowl scroll centrifuge, or it is also called as a decanter centrifuge, which is operated at lower speeds, that is 2000 to 4000 rotations per minute. This type of centrifuge is basically useful for continuous handling of fermentation broths with heavy cell lysates and coarse materials. This centrifuge is more frequently used in the handling of sewage sludge, which contains very high percentage of solids. This is the diagram of this decanter centrifuge. You can see here, this is horizontally positioned centrifuge, uh, and it rotates like this. From this end, the slurry will go inside the rotor of this centrifuge, which is operating like this. So whatever the liquid portion is there, that will run towards this end, that is indicated by the green arrow. So the liquid will run in that direction, and the solids will run in opposite direction, which are shown by a red arrow and black colored solids. So it will run towards the conical end of this rotor, and solids will be discharged from the this end, and liquid will be discharged from this end, that is indicated by green arrow. This is the decanter centrifuge. You can see in this video. So you can see the rotor inside. It is a closed system, and you can see the operation of this. So you can understand very easily. You can see that the feed is going from the right hand side, and it is horizontally continuously it is rotating. So whatever the liquid is there, it is coming out of the horizontal plane, and solids are getting gathered at the conical end. So you can see here, this this is how the solids are getting discharged from conical end, and liquid is getting discharged from this end. This is how the decanter centrifuge it operates. So it is very easy to understand from this animation. Then the next type of centrifuge is called as multi-chamber centrifuge. So as the name indicates, it has a central rotor, and this rotor, it consists of multiple chambers. You can see the arrangement of these multiple chambers. So whatever the liquid which is fed in, so ideally this centrifuge is for the slurry up to 5% solids, of particle size having zero point 0.1 to 200 micrometers in diameter. It consists of a series of concentric chambers, stainless steel chambers, and these are mounted within the rotor chamber. Maximum speed of operation is 6,500 rotations per minute, and it rotates like this. When it rotates, you see the movement of the arrows, black arrows show the movement of liquid. So this liquid, while the centrifuge is under operation, it takes a circuitous route, means it takes a circular route, which is shown by the black arrows. And while uh, the liquid is running inside these chambers, so this blue arrow, it shows that this is the place where the particles or the solid particles are going to be deposited onto the inner side wall of chamber. This is where the particles are going to be deposited inside the inner wall of this each chamber, like this. And whatever the liquid is there, it will be continuously, it is continuously flowing from the, sorry, so it will be continuously flowing, and it will be collected from the topmost end, as you can see here. This is how the disc bowl centrifuge operates. You can see, okay, sorry, this is how it operates, like this. This is a model. Okay, the video for this is currently not available. We'll move on to the next type of centrifuge. It is called as the disc bowl centrifuge. Disc bowl centrifuge, it consists of a central inlet pipe which is surrounded by a stack of stainless steel conical discs, like this. So this is how it rotates in this plane. You can see these are the nozzles for the discharge of solids. That means the solids are thrown out of, means it is thrown along with the wall of this disc bowl centrifuge. And you can see the feed is coming inside, which are shown by the black arrow, and again it is going to take a circuitous route, which is shown by the black arrows. So these lines having slopes, these are nothing but the disc, which are the stainless steel plates. So you can see that the solids will gather along the wall of this centrifuge, and the liquid will be coming out of this centrifuge as effluent. You can see it is mentioned effluent, so that is the end from where the liquid is coming out. So the broth to be separated flows outward from the central pipe, then upwards, and then inwards between the discs, which are placed at an angle of 45 degrees Celsius to the axis of rotation. You can see here these are the plates which are arranged at 45 45 degrees, uh, and the speed of operation is 5,000 to 13,000 rotation per minute, and it is one of the very efficient centrifuges. The only problem associated with this type of centrifuge is the cleaning of the plates is tedious, or it is a complicated job. So it's complicated design and difficulty to clean it. These are the only disadvantages of this centrifuge, and it rotates like this. So this animation, it shows the operation. You can see this the design of this centrifuge. These are the plates. You can see the disc which are arranged at 45 degrees. So the liquid is coming inside. You can see containing the, you can see it contains the unseparated liquid, which is shown by a color code. You can see the clean liquid, which is shown by yellow color, and the concentrate, means the solids, are shown by black color. So you can concentrate how it actually is getting operated. So this animation shows that the liquid is then the solids are then discharged from the bottom. You can see it is gathering, and now it is getting discharged from the bottom. Yes, like this. And the clear liquid, it is coming out here. This is the outlet. You can see here the yellow color coming. So this is how the liquid is coming out from this end, and the solids are getting discharged from the bottom. You can see the way of rotation of this rotor like this. So it clears the operation of this type of centrifuge. This is another video that shows the operation of this disc bowl centrifuge. These are the solids which are entering inside, and these are allowed to thrown away inside the disc, stainless steel disc, and this is how the solid particles are getting distributed through these holes like this. These are the solid particles which are not allowed to enter inside due to centrifugal force and the arrangement of discs. So they will return back, they will bombard onto the disc, and they will return back and fall onto the outside portion here along the wall. So there is a space piston, so which is responsible, which is open periodically, it is open, and the solids are removed. This is how the solids are getting separated. Very interesting video. This is how the solids are getting, yes, there again it is closed after the discharge of solids. This is the movement of clear liquid, which is shown in yellow color. From the center the slurry is fed in, and from the outside pipes the clear liquid is coming out of the centrifuge. It is a continuous process which is responsible for the handling of very high volume of fermentation broth. So that is about the disc bowl centrifuge. So this is all about the centrifugal. Then there is a centrifugal extractor, which is called as the Podbilniak centrifugal extractor. This is basically very useful for the separation of heavy liquid phase from light liquid phase. So it is called as, let us see the operation of this Podbilniak centrifugal extractor. The principle of operation. So this is basically useful for the separation of liquids. This is the Podbelniak centrifugal extractor, which is operated continuously, and it is very useful for the separation of light liquid phase from heavy liquid phase. So this video shows the arrangement. This is the rotor which is rotating horizontally. You can see there are pipes inside as well as there are holes inside. This is basically useful for the liquid-liquid extraction process, the reactions and their separation, as well as for the washing. So the step of operation, it includes extraction, reaction, separation and washing. This is the centrifugal extractor that you can see. Inside there is a rotor and there are stainless steel rotor which contains several pores or holes of specific size and which are arranged like this. You can see these are the holes and these are the extraction pipes which are connected to rotor. So these extraction pipes are for the separation of heavy liquid phase and light liquid phase. So these pipes are inlets and outlets for the heavy liquid phase and light liquid phase. A very interesting design, and basically the design, it makes possible to separate heavy liquid phase from light liquid phase. So this is the lead portion. This is the diagram which is shown in the book of Whitaker. This is the shaft. This is how it looks like. The arrangement is like this. These are the stainless steel disc which contains the holes, and these are the distribution tubes. So the distribution tubes are positioned at various positions or distances from the central rotor. These are called as ASCO tubes. So some of the tubes are attached to the rotor, central rotor, and some of the tubes are attached outside. This is the end for heavy liquid out. This is for light liquid in. You can note that the this is light liquid out and sorry, heavy liquid out, and this is light liquid in. You can see that the light liquid inlet is at the center of this rotor. As we have seen in case of tubular bowl centrifuge, that light liquid phase, it will remain in the center, and heavy liquid phase, it will be at the along the wall of the rotor. So a detail about this, I think we'll discuss more about this in the liquid extraction. So there I will repeat this. For the moment I will stop this video.